Good afternoon, everyone. Today I will be talking about the COVID-19 mRNA vaccines. So to understand how the mRNA vaccine works, I'll go through the differences between DNA and RNA, the processes of translation and transcription, the structure of the COVID-19 virus in relation to the vaccine, and address some of the common misconceptions with the vaccine. DNA, also known as deoxyribonucleic acid, consists of two polynucleotide strands in the shape of a double helix. Each polynucleotide strand contains 4 different nucleotides with 4 different bases: adenine, thymine, cytosine, and guanine. The 2 strands bind together by complementary base pairing, meaning that only adenine and thymine can bind together, and only cytosine and guanine can bind together. As a result, the second DNA strand is the complementary base pair of the first strand. And the sequence of these DNA nucleotides create a gene which codes indirectly for a protein. RNA, ribonucleic acid, unlike DNA, is a single-stranded polynucleotide. However, it also has 4 nucleotides, but the thymine in DNA is replaced with uracil in RNA. It should also be noted that adenine and uracil are complementary base pairs, and RNA is used for messenger RNA, transfer RNA, and ribosomal RNA, which I'll talk about later on. The process of transcription is using DNA as a template to synthesize mRNA. This occurs in the nucleus of the cell and is a 3-stage process. The first stage is initiation, where the enzyme RNA polymerase binds to the DNA, unwinding the double helix and separating the 2 complementary strands. This then allows elongation to occur, where 3 RNA nucleotides bind to the exposed length of DNA. As the RNA nucleotides collect on the DNA strand, RNA polymerase then joins the nucleotides together, forming the mRNA strand. After this, the DNA has been copied onto the mRNA strand. Termination occurs where the DNA, RNA polymerase, and mRNA separate. The mRNA is then used in the process of translation, where proteins are synthesized from the mRNA strand. The mRNA acts as a template for the protein to be synthesized on. And what proteins are that they are a sequence of amino acids which are bonded together. On the mRNA strand, 3 consecutive RNA nucleotides are called a codon, and each codon has a corresponding amino acid. So when the mRNA leaves the cell nucleus after transcription, ribosomes bind to the mRNA. This allows a molecule called transfer RNA to bring amino acids to the mRNA strand. Transfer RNA carries amino acids to the correct place on the mRNA strand. And as the amino acids accumulate on the mRNA strand, they are bonded together, forming the protein. So the COVID-19 virus's structure has a lot of S proteins on the surface of the virus. These are more commonly known as the spike protein. What the spike protein does is that it allows the virus to bind to a host cell, allowing it to transfer its genetic information into the cell to take over the cell's function. As a result, the virus can then use the cell to create more viral particles, leading to a viral infection. And this makes the spike protein a useful target for vaccines and the immune system. So the function of the vaccine is that it contains the mRNA strand to synthesize the COVID-19 spike protein only. So when the vaccine is injected into our body, the vaccine enters one of our human cells. The mRNA is then released into the cell where the process of translation occurs, where the mRNA is then used to synthesize the COVID-19 spike protein. After the protein is synthesized, it is then displayed on the surface of our human cell. As the spike protein is displayed on the human cell, it belongs to the COVID-19 virus. It is identified as foreign to the body, and therefore it is considered an antigen. This then triggers an immune response, where B cells and T cells rapidly produce antibodies with different binding sites to find an antibody which can bind to this antigen, in this case the COVID-19 spike protein. As the correct antibody is found, this allows them to bind to the COVID-19, to the cell, allowing phagocytes and T killer cells to destroy the cell displaying the spike protein. In addition to this, the antibody used to bind to the antigen is also remembered by the B memory cells. So if the real virus were to enter our body, our immune system would trigger an immune response, and the antigen on the COVID-19 spike COVID-19 virus would be recognized, and we can rapidly produce the correct antibody, stopping the virus from infecting any of our cells, leading to a viral infection. And this is how immunity is developed. And this technology has large potential, as it does not use the live virus itself. Rather, it only codes for an antigen, which is harmless to human cells, to trigger an immune response to develop immunity. So a conventional vaccine is very different to a mRNA vaccine. What the conventional vaccine does is that it contains live virus, so a weakened or inactivated disease-causing antigen to trigger an immune response, to train the immune system to find the correct antibody. In comparison, the mRNA vaccine simply provides the code for the spike protein to be synthesized, creating a replica COVID-19 virus to train the immune system in that way. Furthermore, mRNA does not actually contain any of the virus's genetic information, so when the spike protein is displayed on the human cell, it is not considered as infectious or dangerous to our bodies, as it is still essentially a human cell and doesn't contain any of the virus's genetic information. Furthermore, the mRNA strand cannot enter the cell nucleus, as translation occurs outside of the cell nucleus. So this means that the mRNA strand cannot damage or modify our DNA in any way. And finally, the mRNA strand, after it has been used to translate the COVID-19 spike protein, it is then broken down in the cell, stopping it from continuously producing this antigen. And yeah, thank you for listening.